Alrighty, Unit 3. We are going to be talking about the cell membrane. This can be found in eukaryotic and prokaryotic cells. Eukaryotic and prokaryotic are just two different types of cells. We are eukaryotic if you need a reminder. So let's get started with Unit 3. Unit 3, cells are made up of a cell membrane. This is the outermost layer of a eukaryotic cell. If you are a plant cell, you probably will have a cell wall to help with the structure. But our cell membrane is in charge of moving things in and out of the cell. There's going to be two types of ways for a cell to do that, all right? Before we get into the two types, I actually want to draw out what this cell membrane looks like. It's going to be a bunch of phosphate heads. It's a bilayer, so there are going to be two layers working together. And the fatty acid tails. So we have one right there. So what I'm drawing is two coming out of this one, two coming out of that one. As a whole, they just look like a little bridge, okay? And a cell membrane, you're also going to see, so this is what our cell membrane looks like. This part right here, that is going to be made up of lipids. The majority of the cell membrane is going to be made up of lipids. This green thing is going to assist the cell membrane, the lipids, and this is called a protein. Cool. So our proteins are going to be helping the cell membrane. Why does the cell membrane need help in moving things along? Well, you have small molecules, which is going to be water, carbon dioxide, oxygen, or you have large molecules, okay, like salts, potassium, that cannot go through this small lipid membrane, okay? They need help from the proteins. So jumping in, we have two types. We have passive, let me zoom in a little. There we go, passive, and we have active. All right, so there's going to be three types of transports for both of these, what do we call it? Transport, sorry. We have diffusion. We have facilitated diffusion. So facilitate, remember, it means to help. I'm going to put DIFF, just in short. And then we have osmosis. I'm going to draw this in blue, and we're going to get to that in later. Now, what do all of these have in common? Well, all of these have in common the fact that they do not require energy. Let's move down here. No energy. So energy, also known as ATP. And they go from a concentration of high molecules to low molecules, okay? When I say concentration, I mean the amount of particles in one space. So it's going from lots of particles to less particles, solute, right? So diffusion, that's going to be small molecules going through the lipid side of the cell membrane. Facilitated diffusion is going to be our protein, all right? That's the friend that's on the VIP list that can get you into the club, all right? They're helping, our proteins are helping, okay? On the opposite, or actually we're going to continue. I was going to compare it to active, but we're going to continue. So facilitated diffusion uses proteins help to get through because they are large molecules. Osmosis, I'm going to make a separate little bank over here for osmosis, is the movement of water. Okay. Osmosis, all right. The movement of water. It's still going from a high concentration to a low concentration, but there's going to be a few environments that you're going to see cells be in. So, and there's 3 specifically. This is going to be, let me mark that in red. This is our cell. We have 3 situations or environments. So we have one environment, you have a lot of red things. What do those red things mean? All right. Those red things is what we call solutes. What is being dissolved or dissolving, right? You have solvent, which is the water, which is doing the dissolving, okay? In the situation where you are acting as water, you're gonna go where solutes are the highest. Let's say solutes are party, all right? You're gonna follow the party. And so if you are following the party, you're gonna go outside of the cell. Party's outside, so you're gonna go outside, okay? If you're outside, you'll find a lot of active people, so people who are hyper. And so that reflects our environment. It's hypertonic environment. There's a high concentration of solutes on the outside. Okay? You will have environments where there's a high concentration on the inside. Maybe it's cold outside. Maybe it's cold outside and you want to avoid getting hypothermia. That's just being really cold. So you're going to stay inside. If you're water, you're going to go inside or outside? Inside. You're going to follow the party, okay? An easy way to remember what this solution is going to be the hypothermia. You're trying to avoid hypothermia, so you're going inside. Hypo means low, all right? Hypo, and then hypertonic. So there's a low concentration on the outside. And then we have what we call isotonic, where the concentration of molecules is going to be the same within and outside the cell. All right. So again, this is the movement of water that falls under passive transport. It does not require energy, and it's going from a concentration of high to low, right? Our water in osmosis, remember, it might be confusing because you're like, well, if the concentration is outside, you want to go inside. Remember, we're talking about the movement of water, and these red marks represent solute, but water is a solvent. Oops, water is a solvent. Okay. So moving on to active, we have 3 types as well. Let me change my font size. We have what we call pumps, exocytosis, then endocytosis. All right, so these 3 are similar because they require energy. Why do they require energy? And remember, ATP is energy. They require energy because they're going from low concentration to high concentration. So that's not really favorable. You don't like to be shoved in tight spaces with strangers, right? That makes you uncomfortable, but sometimes Energy is going to come in and they're going to push you against what you want to do. Okay. We have, let's start with our exocytosis and endocytosis. Exocytosis is going to be when something, let me draw a cell. So exocytosis, something is leaving the cell, okay? Exo, exit. Let me scroll up, there you go. Exo means exit, okay? Things are leaving. So this might be waste, all right, that the cell produces, and I'm gonna draw it in brown to help you remember that. Waste, okay, exiting. Your cell doesn't want any of that, and waste is gonna pile up on the outside of the cell, preferably, okay? So erase that. Unnecessary drawing. Okay. Next, we have endocytosis. You can remember endocytosis because you're entering the cell. Okay. You're going into the cell. You can be food. You can be, yeah, we'll draw food. Pretend that's a hamburger. So it can be processed by the cell. The cell needs food. Okay. Nutrients. Finally, the final type of active transport is going to be, oops, it's going to be pumps. So let me redraw our lipid lipid bilayer, phospholipid bilayer. This one also has a protein. This protein is going to be pushing things from low to high using energy. And often we'll see that within a sodium-potassium pump. What does that mean? Our cells are producing a bunch of sodium. They're producing sodium, not a bunch. And these proteins are going to help push them against the concentration. So it's highly concentrated outside, but the cell doesn't care, okay? It just says, meh, I don't want you here. Get out. That's going to be our sodium, right? Then we have potassium. That's highly concentrated on the inside, and our protein is going to bring that in, regardless if there's not that many outside, all right? So those are the three types of active transport that the cell undergoes. This is what a cell looks like. You'll have our nucleus, you have an endoplasmic reticulum, Golgi, all its little organelles that make it living, all right, that help it live. We just talked about the cell membrane, the outer part that helps transport things in and out, okay? And if you zoom in, this is what it looks like. You have a bunch of little phospholipid bilayer. There's 2 layers. You have occasional proteins, all right? And things are going to come into the cell in 2 ways, either active or passive transport, okay? Diffusion, facilitated diffusion. Notice that we have proteins both on facilitated or on passive and active, okay? Facilitated diffusion is when the cell is moving things in and out without moving things from high to low without energy. And then we have pumps that also uses proteins, but it's moving from low to high. If you have any questions, please feel free to ask me, email me. That's going to be it for Unit 3. So hopefully this helped clear some air. You guys have a great rest of your afternoon, and I will catch you guys later.